All right, let's move on with chapter 16. Actually, lecture 16, but not chapter 6. We're going to return to chapter 6 in a couple of videos. But for now, we've got two videos that are going to take place in chapter 11.3. So, if you have time, I recommend looking over the book and just looking over chapter 11.3 right now. I have specific instructions about which places not to look at. I want you to have these ideas in your head, and then I can talk about them more effectively. So, jump to section 11.13, and let me just check. Okay, we do have actually a microphone working. Good. So there's actually a dish apparently called the dancing squid. I haven't ever seen this. There's probably videos. I'm not sure I want to look for them because it seems a little bit weird, but it seems cool too. So squid is placed on a bowl of rice, and then you add soy sauce to it, and the mollusk starts to move. It starts to dance because the nerve fibers of the squid, they're still intact, and they contract when you actually add salt. So why do nerves react to salt so much? It's because they run on salt. And the way that they work is they actually are doors for salt. So this is actually, the way that nerves work is they work on transporters that involve moving salt ions across a membrane. They are the doors in the membrane for salt. And the main reason I want to talk about them now is because they act exactly like enzymes. Everything that you learned from chapter 6 is applied in chapter 11.3. So I'm like, why not talk about chapter 11.3 now? Here it is. So they move the ions in and out of the cells. Because they are enzyme-like and they work with kinetics, we can use our enzyme kinetics tools to quantify how they work. So overall, there's three types of transporters. There's a simple one that just opens up and lets something through. That's the substrate, and it's a uniport that moves something from outside to in. Then there's two kinds of co-transporters. Sometimes you can get a two-at-once kind of transport. There's symport, in which both substrates move through in the same direction. Then there's antiport, when one substrate moves in and one substrate moves out. These are useful because you can pair two motions together, and one of the motions can be energetically favorable, and the other motion can be energetically unfavorable. But when you add them up, it adds up to be favorable enough to work and move forward. That's why you have symport and antiport, because you can actually pay for the energy of one with the other one. So we're going to have to think about energy. A good thing is we have a term for talking about it with delta G. Sodium channels are big, huge transmembrane proteins. In biology class, you can geek out on all the structure of this. Here's a little bit of the structure. This structure doesn't matter that much to us. The only thing that matters is that, number one, they're big and complex. They're embedded in the membrane. They have a hole in the middle. And then they have a way to filter what goes through the hole. So there's a selectivity filter at the top of the membrane that selects which ions can enter the door. This is the important concept, that you have the selectivity filter on top of it. Then you have the hole that goes through. And then you have one more thing you need to know about, which is called the activation gate. The activation gate is a helix that can actually activate, open up the door, and close the door. So it's an activation gate, and there's another helix that's a voltage sensor. These are embedded in the membrane. We're going to talk more about how these things work, but they have alpha helices all through them. And the alpha helices can do different things. Let's look at what one looks like. That was sort of the secondary structure of one. Here's the tertiary structure. But really what it is just a big hole with a couple of different areas. Has a selectivity filter in pink at the top. Has a central cavity in the middle that doesn't do much. Has an activation gate at the bottom that can open and close, physically opening and closing the hole for things to go through. So just remember, selectivity filters at the top. That sometimes is important to some homework questions. What's really cool is these have a selectivity filter that selects for sodium. Sodium gets in and out of the cell through these, and sodium is in salt. This is why salt will make nerves twitch, and why salt in your wound will make your nerves hurt. So sodium transport through these channels is how neurons work. sodium will flow in spontaneously because sodium is naturally low inside the cell and it's high outside the cell. So because you have this concentration imbalance, when you open it up, sodium spontaneously will flow across the membrane. There's a negative delta G for sodium moving from outside the cell to inside the cell. All you need to do is open up the door. The reason why you just need to open up the door is because over on the other side of the cell, you've got a protein that's burning ATP all the time that is pumping sodium out of the cell. It's burning ATP to create this imbalance. Because it does so much work, the sodium channel just has to open up and let the sodium do its thing, flowing down the concentration gradient to the negative delta G. The reaction happens spontaneously as a result. Oh, and the other cool thing, the voltage sensor actually is just a charged residues on an alpha helix, and what will happen is when the membrane reaches a certain charge imbalance, it's always imbalanced. You always have more positives outside and more negatives inside. So normally the helices are pulled down because they are positively charged. They're repelled by the more positive outside. They're attracted to the more positive inside. In that configuration, the door is closed. But when the nerve fires, the nerve depolarizes, and that means that the outside and inside aren't as different anymore. Therefore, the activation sensor can move up a little bit. The difference between the plus and minus is not as great. There's not as much plus on the top. Therefore, the pluses in the activation gate or the voltage sensor can move up and the activation gate can open. It's a very straightforward mechanism. You do have to keep track of charges. And again, we're not focusing so much on how this works. It's really cool. It's a topic for another day about exactly how nerves work. But the one thing that we want to say is, okay, how do we know how much energy can we get out of this? This is the equivalent of the delta G of a reaction. If the delta G of an enzyme is substrate versus product stability, the delta G of a transporter is sort of outside versus inside solubility or concentrations. And that determines the delta G of moving from outside to in. So, if you just have an uncharged solute, any kind of solute, what matters for whether it's going to move in one direction or another across a membrane when you give it a chance, if you have two containers and one of them is full of substrate and the other one is less full, you will have an osmotic pressure. If you allow them to go through the membrane, they're going to more often go to the right than they are to the left, just because there's more on the left and they're going to sort of push through to the right. Just think about this. This is what dialysis is based on. This is the normal situation. When you have an imbalance of concentrations, things will have a negative delta G for moving from the higher concentrations to the lower. It's just the way concentration works. They will if you keep the door open, they'll keep moving until eventually the movement in and out will equalize because the concentrations are equalized. Once you have equal concentrations, it's just as likely to move to the left as to the right. You've reached equilibrium and you no longer can do kinetics experiments. So the delta S is spreading out. When you move from left to right in this before equilibrium one, from higher concentration to lower, you have a negative delta G. It's spontaneous, it happens naturally, and you have a positive delta S. Think about it. You have the solute is spreading out. Once you reach equilibrium, you have a delta S is 0. There's no difference in spreading out from left to right. There's no difference in concentration. There's also no difference in delta G. The living cells are the situation on the left. So by the way, if you ever talk about osmotic pressure in a biology class, be a little aware, if you're the kind of person that likes to challenge what the teacher says, there's a lot of misconceptions that are taught about osmosis. And I have a paper at the bottom that's about exact misconceptions about it, about how osmosis works. We don't have time to get into this now. We can actually talk about this in capstone class if you're interested in correcting misconceptions. But for right now, we're not really talking about the biological thing of how does osmosis work. We're just taking for a fact that osmotic pressure is a thing. Osmotic pressure is, if you're moving from C1 to C2, it's going to be a negative delta G. And all you have to do is open up a transporter, and the delta G will determine how much can move before it gets to equilibrium. The other factors will determine how fast it moves, the kinetics of diffusion. So the one thing about diffusion is you have the transporter down here. So you see it down here. And the transporter catalyzes the movement of the solute. If you think about it, there is a possible movement of the solute. Let's say that you have a sodium ion on this side of the membrane. It can move across the membrane. It's just not very likely and not very fast. Because it's surrounded by water, it's entirely happy being surrounded by this polar material, right? To move it across the membrane, you have to strip off all the waters. You have to desolvate it. That takes a lot of energy. There's a big energy mountain. Then it has to be in the middle of the membrane where it's unstable and unhappy and high in energy. It's still on the top of the mountain. And see, it's a red line right here. If you have the desolvation, which is this little hump, the top of the mountain there. Then you have it in the middle of the membrane. It's not happy. Then you have to resolvate it, which involves moving the waters around in the exact opposite way that you took them off. And then finally you're inside the cell and you can return to your delta G, and the delta G is determined by the concentration. In this case, the concentrations are equal on each side, so it's kind of like this shows you the height of the mountain of the uncatalyzed diffusion reaction. What the transporter does is it lowers all three of these transition states. It binds to them. And so it binds to the ion in the middle of the membrane, but it can bind to it with charged residue, so it's much happier than it would be. It will move through much faster. It has a space to move through. And so this is exactly like the way enzymes bring down the transition state by binding to it. It's just here the transition state is in the middle of a membrane. Now, this is a passive versus this is passive, and this is dependent on the concentration of which way it will go. You can also stack the deck. You can use ATP to sort of power an unpopular or disfavorable kind of movement. You can move something against its concentration gradient if you pay for it with a bunch of ATP. So that's solvation. By the way, remember that I said that would be a term that's important. You have desolvation on the left, resolvation on the right, and that accounts for part of the energetic penalty you have to pay because you have to strip those waters off in order for the thing to get across. The transporter can help make that process easier. So the way that this transporter works, here's a transporter called GLUT1. And it's a glucose transporter. It basically means it has a pore that's the right size and charge to fit glucose. It's actually pretty easy to make a glucose transporter, and anybody with diabetes is very interested in how these things work. GLUT1, to make a simple kind of glucose transporter, that's what GLUT is what it stands for. All you have to do is you have to make a helix with this pattern: serine, leucine, valine, threonine, asparagine, phenylalanine, isoleucine. And if you look at that, that makes a helix wheel where all these polar residues are on one side and all these nonpolar residues are on another side. Well, if you end up with 4 of those helix wheels, you end up with this situation where you can arrange them and you can make a whole little wall out of your polar residues, and you can have the outside of the transporter be all hydrophobic stuff. This dissolves in the membrane. This creates a polar pore that can bind glucose, and glucose is very happy to move through that hole in the membrane and to move inside. This is a simple way to make a transporter, and just see how easy it is. It also involves the helix wheel concept, which I want to review. You basically take 4 of these, stack them around each other, and you've got a glucose transporter. Very cool. Here's a structure of a glucose transporter. It's actually a xylose transporter, which is another sugar. That's in the next chapter. But I want you to see, It's basically a bunch of helices. And the other thing about the helices, look how they form a V. There's a V where they go up and down. And it turns out that the V is sort of open to the sugar. The sugar can go in to bind to the middle of it, and then it can actually close up and it can form like an A. And then it lets the sugar through. It's a little bit like an airlock. It lets the sugar in, closes the top, opens the bottom, and lets the sugar through. This right here is with it open on the top. Kind of cool. And also useful for you as you think about protein structures. So we can come up with a kinetic mechanism for how glucose moves through. We can even come up with different steps. We can say that we have the open step, we have the binding step, then we have the conformational change where it closes up, then we have the conformational change where it opens up at the bottom, and then the sugar release step would be another one. We can draw arrows for all that. We won't do this that much, but I just want to show you the principles of enzyme kinetics can apply to this as well. And so that's when we move on to lecture 16.3. Now, this was recorded previously. I'm going to introduce you to it. And so I might use different words for the worksheets, but they're the same worksheets that we talked about. There's one for each lecture this week. And there's homework 6B is going to include homework problems from this. And so we're going to start off when you move on to the next lecture. It's going to start off with this, where we have the equations for the kinetics of transporters. We showed you how the arrows right here, we could do a glucose transporter. We could come up with the arrows for the different movements. Well, it turns out we can come up with all the arrows that we come up with. We've already done the work. We've already done the math, because we can do the exact same arrows as we did for enzymes and substrates. In fact, this is the 4 or the 6 arrow situation that we talked about when we first talked about enzymes and products and things like that. And we said that we can write these arrows. This is exactly the same. It's just that it has S out instead of S, T instead of E, and SN instead of P. If this is all true, then all we have to do is take out our Michaelis-Menten equation here and switch the labels around. So there's more about that in the next lecture. You probably want to think about that and let this sit, and I will be posting all this very soon. So once you finish those other 2 lectures, they are shorter, and we're getting shorter and shorter each time. You'll have the equivalent of a Friday lecture where we are asynchronous and recorded, but we're still going through the biochemistry. And I'll see you on Monday to talk about sugars.